Thank you for agreeing on my another tricky marketing efforts. So, sorry, I forgot to introduce
All right.
myself. My name is Alina Bonadonna, and I will be a moderator of our today's webinar. And guys, I see already a lot of attendees here, and just shoot on reaction on the left button that you are see as well, hear as well. I will start first, put my heart on. So waiting for more hearts, waves. Great. Looks like people see as well, hear as well. Amazing, amazing. Great. Cool. So, just a small guide about how we will go on today. As always, Julian will take his 30-40 minutes of presentation. After, he will answer all your questions, but of course you are able to type it in the question window during the presentation as well. And pay attention on the upper right corner where you can see attached materials about our new test, which we will present today. So guys, are you ready? Julian, are you ready?
I'm ready, yes.
Okay, great. Let's try to share your screen and then I will disappear for a while.
Yeah. You need to give me the permission to share.
I think you should be fine. Okay, I make you as organizer. Yes. So now you can do all possible manipulations.
Where is it?
Great. Guys, just let me know if you see materials attached.
So
Just
see everyone sees the file, the
ye-
screen with the
yeah, yeah,
slides.
yeah, yeah.
Okay.
So, Julian, I disappeared. Time is yours. Enjoy.
Okay. So hello everyone again. It's a pleasure to talk to you today and present our newest product, the N-Light Listeria SPP, and also to tell you something about how Listeria SPP detection is used in environmental monitoring in food factories. So first, I would like you to introduce to you some basic facts about Listeria SPP. So the genus, so the name Listeria is known since 84 years already. A scientist called Mr. Peary introduced it in 1940. And for a long time, only one species was known as this is the famous Listeria monocytogenes, and before it was called Bacillus monocytogenes. But Listeria as a own group of organisms is called like that since then. At the moment, there's like 28 different species of Listeria described and published. But from time to time, there's new ones discovered in nature, and then there will be another one described. I'll tell you later which are the most important with respect to food factories. Many of you probably know also the names of it. In generally, Listeria, so all Listeria are gram-positive, rod-shaped bacteria. On the right, you see an electron micrograph of L mono. And they're mobile also, at least under certain conditions. And you see that because it has flagella, you see nicely in this electron micrograph. And also important with respect to sterilization is that they're non-spore forming, other than other gram-positive bacteria. Then they have a very flexible metabolism, so they're facultative anaerobe, so can grow both with and without oxygen to fermentation and oxidative metabolism. And also important with respect to food production and shelf life of foods is that they're capable of growing a really wide range of temperatures. So starting from 4 degrees, so refrigeration temperatures to up to 40 degrees. And that's really a problem because that means that even if you store your foods refrigerated, um, often Listeria monocytogenes can multiply if introduced into the food during production. Another thing which troubles food manufacturers is that Listeria in general are very resistant, except for heat. But a lot of other stresses like high salt levels, salts are often also used as preservatives, drying, acidic or basic pH also often used as a preservation measure in foods, antimicrobial agents, quite a few antibiotics they are naturally resistant to. The good thing is, though, for foods is that it can be killed by pasteurization, and pasteurization is defined, for example, as 72 to 74 degrees heating for 15 seconds. That's the ultra short pasteurization, or a batch pasteurization would be 36 to 65 degrees for 30 minutes. So both of these processes are used widely in food industry, for example, for milk. Then the last basic fact I want to mention is two of the most characteristic enzymes which are found in all Listeria species is beta-glucosidase and also catalase. So where do the Listeria belong to? Already 1991, the first phylogenetic tree was published based on sequence data on the 16SR RNA data, and it was found that the Listeria species known until then really closely clustered in one region of the phylogenetic tree, and they belong to the so-called low GC content gram-positive bacteria. Close relatives of Listeria, for example, bacilli, so Bacillus cereus group, Bacillus thuringiensis, Bacillus subtilis, which are also very common, for example, in soil. Then also quite closely related are lactic acid bacteria like Leuconostoc or Lactobacillus, Streptococci, another group of lactic acid bacteria, the Lactococcus lactis, the famous ones used for yogurt. And then also the Enterococci, they're closely related to Listeria. So here is a more close-up view of only the Listeria SPP group of bacteria, a newer genetic analysis. Actually, it's based on the amino acid sequence of 325 different genes, which are found in all of these species, in all of these strains, and then you can build a really highly precise phylogenetic tree for these organisms, and then you see a clear grouping of a specific group of Listeria called Listeria sensu stricto. This is the most important group related to food safety, so it includes Listeria monocytogenes, Listeria innocua, Listeria welchimeri, Listeria ivanovi, and Listeria saligari, and Listeria marthi, but this one is very, very rarely found and actually has only been found in 2 places yet, so it's not really relevant. And then there's a broader group of a bit less closely related Listeria. They're called Listeria sensolato, so Listeria in a broader sense, which includes strains like Listeria, species like Listeria weihenstephanensis or Listeria rocurtiae and others. So why is Listeria species in generally of any relevance to food and to public health? It's mainly because of Listeria monocytogenes, because this is a human pathogen. It can infect humans, but also some warm-blooded animals. And this is the only human pathogenic species in Listeria spp. And then you have a second one, Listeria ivanovi, but this is only infecting animals and very, very rarely humans. So it's really all about L. mono. Listeriosis, that's how the disease is called, which is caused by Listeria monocytogenes, is a very dangerous foodborne illness. In Europe, for example, there's data from 2017. It's 2,480 confirmed cases, invasive human disease, 225 deaths. So quite, if the disease is really happening, quite high fatality rate. For the US, it's estimated approximately 1,600 cases and 260 deaths per year. This hasn't changed much the last years. I think it has even gone up a bit. The thing about this disease is that there are specific groups of the population which are specifically vulnerable, and that's, for example, pregnant women, which are 18 times more likely to become ill. And what is even worse is that the fetus in the women can also be infected by this if the mother is sick. Then, which is a growing group of people, certainly, is elderly people above 65, and also other immunocompromised people, which have to use, for example, immunosuppressant medicine and so on. They can also develop severe infections with Listeria monocytogenes, which can then lead to sepsis and meningitis, and these conditions can eventually also lead to death. That's because it's such an important pathogen, and it's really almost exclusively foodborne. There's in most countries in the world there's limits for Listeria in the food, but usually these limits are really only for Listeria monocytogenes. So usually there's no like limits for Listeria spp as a general group of organisms. For example, in Switzerland, ready-to-eat foods, which can support the growth of Listeria monocytogenes, there's the rule that no L. mono must be detected in 25 gram when the product leaves the factory, and less than 100 CFU per gram at the end of shelf life. On the other hand, RTE foods which do not support growth of L. mono, they can have 100 CFU per gram when the product leaves factory. However, it's specifically mentioned in the regulation that if the food is to be used for babies or for specific medical needs or for immunocompromised persons, then there should be no L. mono detected in 25 gram of food. Where do these Listeria bacteria occur? So there's been quite some nice studies out there and reviews. You can see also the citation here if you want to look into more detail into this. It's found very broadly all across the world, in the soil, in agriculture environments, on farms, in the wilderness, in decaying plant matter, in rural waters, urban runoffs, so almost everywhere, in irrigation water and so on. So this is mainly data for L. mono, which is from public health the most relevant, but in a few cases also L-SPP, and usually the numbers for L-SPP, which in the end include L. mono, are higher than for L. mono, which makes sense because that's then a more broader set of microorganisms. And the ratio, so the percentage of samples which are positive can be quite high, up to 20% or 30% even. If you go to tropical countries like Nigeria, you can even have up to 78% of soil, water, vegetable samples positive for Listeria spp. Although it always depends on the methods that has been used in these studies, so how well really it has been confirmed that it's really Listeria spp. But in general, it can be said it's occurring very broadly all across the globe. So how do the Listeria spp enter the food factories? So because they're so widespread, and what's an additional problem is that many wild and domestic animals and humans can also be carriers of L. mono and Listeria spp without knowing it, because they will get sick, but still can shed these bacteria over prolonged periods of time. Then it can be brought in with the shoes, with the workers, with pests, insects, mice, then with the raw product, the raw meat, the slaughtered animals, just on the surfaces of fruits, of vegetables, fecal contamination. So there's like thousands of different possible routes where how it can come in. It can also then be on the outside of transport containers, be on the dust, and so on. So actually, you cannot 100% probably prevent that from time to time some Listeria spp will enter into a food factory. The other issue is that Listeria then, once they've entered, can colonize food factories because they have some good growth condition there. If it's wet and if there's like nutrients coming from the food itself, they can colonize surfaces and then form biofilms, either monospecies, but probably in most cases multi-species biofilm. For example, it has also been shown that L. mono can sit in or colonize Pseudomonas biofilms. So what is then the frequency of different Listeria species in food and in food industry? Well, it's really restricted to only a few species which are really found there in such samples. So mainly L. mono and then innocua, the two most important species. And the third and fourth are Listeria welchimeri and Listeria zeligeri, and all the others are only rarely found. So, and the proportion of innocua, mono, and welchimeri, they can vary from 60 to 40, and so there's in different studies you find different ratios. But yeah, these four species, I would say, and especially mono and innocua, these are the most important ones. So how are Listeria detected? I want to go into more detail here into the most important cultural method, which is used in Europe. So that's the ISO 11990/1. That's for the qualitative method. So you first have an enrichment in so-called Fraser broth, half Fraser broth at 30 degrees, so which is not yet fully selective. Then you do a sub-cultivation, another enrichment in full Fraser broth with the strictest level of selectivity. And then from both enrichments, you will do a streak out on a chromogenic Listeria agar. One of the most well-known one is the so-called Aloha agar Listeria, according to Ottavoni and Acosta. And here you have to cultivate for 48 hours to be sure, because sometimes the colony will appear quite late. And on the right, you see such tubes from the Fraser enrichment. So often if there's Listeria growing, but also other organisms can do that, and then you will have such a typical black coloration, which is coming from beta-glucosidase activity. There's a certain compound which is degraded and then reacts with ions and gives these black colorations. And if it's a sample without any Listeria or which is without any bacteria or sterile, probably this broth will stay yellowed. And then the ISO method then calls for once you have colonies with the typical color observed, it's only presumptive, and then you need to do a confirmation by either biochemical test, MALDI, PCR, immunoassay, and so on. So the Aloha agar, it has a substrate for this beta-glucosidase. It's called X-beta-D-glucoside, and so-called indoxyl substrate, and it will give such a blue color, blue-green color, typical for Listeria spp. And only if, in addition to this blue-green color, there's a halo around it, then it's L. mono, because this halo is indicative of phosphatidylinoacetyl-specific phospholipase C. That's a pathogenicity factor of L. mono, and only L. mono and a few other organisms show this activity. However, once you see such colors, it's only like presumptive identification, and you need to do a confirmation on colonies with PCR or something else. And then today's it's really important also now to, once you have such isolates which are confirmed, L mono or Listeria spp, you do whole genome sequencing, and that's done so that you can trace back, like you found contaminated food and you want to trace it back to a specific factory or check where it's coming from, if it's always the same strain. And also, if you have clinical cases, you try to trace it back to certain foods and food producers. I want just to briefly mention the multiple methods for Listeria SPP detection. So very often used method is qPCR or real-time PCR, the other name for it. Also here you need to do an enrichment for usually 18, but also up to 30 hours with a Listeria specific broth. Then there's a DNA extraction step from this enrichment culture, and followed by a qPCR run where you have specific primers for either Listeria SPP or then also L mono. And what you then see is such amplification curve as you see here. This is from our own lab with isolates we obtained from field studies. And you see here the blue line is the negative control, so sterile sample, and then the four different colored lines are all colonies from these aloha plates, and they are clearly positive because at some point you will see the fluorescence go up, which is indicative that there's DNA amplified with these primers. Then also quite well known are immunoassays, for example, the bioMérieux VIDAS assay, where you also have an enrichment first and then do a ELISA, enzyme-linked immunosorbent assays. This means that the Listeria spp cells are captured then with antibodies, specific antibodies washed, and then second antibody, and then a substrate is added, and finally you will have either fluorescence or color readout. So now let's come to our new test. So we have a specific proprietary technology which is called AquaSpark. These are luminogenic substrates, meaning that these are molecules which will emit light once an enzyme cleaves off an enzyme label group. Our test also involves first an enrichment steps, similar to all the other methods. And we optimize the broth for rapid growth, and that's at the same time high expression of diagnostic enzyme, and at the same time suppression of competing microflora. And we achieve this with a specific bacteriophage mix, also a certain set of salts and antibiotics. The enrichment is done in such small tubes in a dry block heater. Then once the enrichment is finished, we do sensitive detection with this AquaSpark substrate. You see here, you see here to the left how such substrate in principle look. You have this enzyme labile group which can be cleaved by the enzyme, and then a specific highly efficient dioxetane molecule which will then spontaneously oxidize and fall apart, decay in the broth, and then there's light emitted from the energy released by these chemical reactions. And this light can be measured very sensitively with a luminometer. We have this small benchtop luminometer, and then you'll get out a numerical RLU signal. And once this signal is above a certain threshold, we call out then the test as positive. So the principal procedure looks as follows. So the swabs, which we also supply, is moistened in sampling buffer. Then the environment of the factory is sampled. The swab is broken into the tube. Then the biosafety cap is closed so that you cannot open it anymore, and no enrichment broth can come out of it. And then the tube's incubated 24 hours, 37 degrees in a dry block heater, and then followed by luminescence reading. Optionally, you can then send the tube, which is closed with the biosafety cap, to a biosafety level 2 lab and do a follow-up analysis, for example, with the ISO method or with PCR to see if you really have Listeria spp or even L. mono. So that I can show you that the test works. We did quite some experiments in the lab, also in collaboration with the Zurich University of Applied Science, where we had a joint project on this. So you see here, we had tested 90 L. mono strains of our strain collection, many of them coming also from food factories, and 98% of them were positive in our test. Listeria innocua, we had 14, all of them positive. Listeria welchimeri, 3 only, but also 100% positive. And this one mouthy strain positive, and also not further characterized food isolates. So where we only know it's not L. mono, it must be some other SPP species, 75% were positive. Then the exclusivity is also very important in tests, certainly that you don't have too many false positives. For the beta glucosidase-based test, like the Fraser broth, there's a big issue that a lot of almost all Enterococcus strains are positive for this enzyme, but luckily we found that for the enzyme we use, it's only 8% positive. Also Staphylococcus, which can also occur in food factories, a small fraction only positive, and also very important in the food environment, the lactic acid bacteria, none of them was positive. And also the Bacillus strain, Bacillus cereus, and so on, none of them positive. Gram positives, also we don't have an issue, none of the common gram-negative bacteria occurring in food factories was positive. Then we also did a study with swabs really on stainless steel with dried cells, and tried to go down to quite low numbers of bacteria inoculated on the surface. So this means you grow the cells and then dilute it down and put it on a plate and wait a couple hours until they've dried. With this procedure, only like 10% usually of the cells will survive. So if you inoculate like 74 cells, there will be only 7 viable cells present once you sample the surface. And here we found also that it works quite well. So most of swab tests we did were positive. 4 of 4 for L. mono at 70, for the one L. mono strain at 74, inoculated innocua, from very low to rather high numbers present on the surface was in all cases except one, there was 5 of 6, but that's still quite good, was positive. Also for Listeria in welchimeri, the same picture, can go down to very low levels and still get out positive results. And as a negative control, it was Enterococcus strain used, and or a sterile control, and none of these was positive. And then a competition assay when Enterococcus fecalis was co-inoculated to the surface together with innocua or welchimeri. Also in this case, all of the tests were positive, so that to us looked quite good. So that's why we also went to field studies, field trials, and we did with our new test some environmental monitoring in an ultra fresh foods factory, 70 samples during or production or equipment after production, before cleaning. And here we got 22% positivity rate, and 5 of 16 of these tests could be then confirmed in subsequent ISO and qPCR analysis. And the positive ones were mainly found on floors, ceilings, drains, and so on. So places where you really would also expect that you may find some positives. In this case, no L mono was found with the ISO method. Yeah, and then in a second trial we did in an ice cream factory, sampled 70 places. Out of these was 39% positive, and here more than half of the positives could be confirmed by ISO and qPCR. And again, positive samples were mainly floors, but also machine parts or the outside of tubings where you have some condensation waters and drain surfaces and so on. And also in this case, no L mono was found. In a meat factory producing ready-to-eat foods. Sorry, there's a mistake. It was only 35 swab samples. Sorry about that. Of this was 26% positive, and 5 of 9 of these could be confirmed later on as being Listeria spp. And here it was machine parts or the feet of the machine, and also a balance and such places where positives were found. And here, interestingly, 2 was later to be found L mono, and 3 was non-L mono Listeria spp. We are currently also sequencing these strains to see which Listeria species they are, but usually we would expect that most of them are like Listeria innocua. So as a summary, Listeria spp is really a very well-defined phylogenetic group, which includes the really important foodborne pathogen L mono, and all the Listeria species cluster tightly together in one part of the phylogenetic tree. The Listeria sensu stricto, which are even more like closely related and cluster in one part of the of these Listeria genus, they are really the most relevant Listeria for food safety. You have already well-established culture-based and molecular methods available of a quite high diversity, which are available for detection of Listeria spp, and as part of that also Listeria mono in environmental monitoring. And now we developed a new luminogenic swab test. Actually, I think it's the really first luminogenic test for Listeria spp, which is compatible with our instruments, and also compatible with a novel device we developed for larger surfaces. I will talk about this new device also in another webinar coming up. This test, on the one hand, it's lab-free. You don't need a biosafety lab. It's safe because we have this biosafety cap, which prevents any spillage of enrichment broth. It's robust, fast, and can easily be done by any factory worker after a short training. And then the good thing is also that it can be incorporated in wider testing schemes. So that means that you can send it out to laboratories for confirmation by cultural methods and also directly by molecular methods. Yeah, and yeah, that's it. And I'm really interested on any questions you may have on these things I presented.
Thank you
Thanks a
so
lot
much,
for your
Julian.
presentation, and thank you for listening.
Thank you so much, Julian. And guess what happened? Of course, we have already some few questions. And guys, please
Okay.
don't hesitate to put on some reactions on this amazing presentation. Like I don't see your reactions, guys. Yeah. Look, Julian. How many hearts
Okay.
you
Okay.
got.
Okay.
Great. So just as a reminder, if you want to get this presentation, just drop some small message to my email, and I will definitely share it. I hope Julian doesn't mind.
No, no, I'm fine with it. Just I'm happy to share it. Yeah.
Yeah. So the first, actually, two questions are coming from Gregor Bogatko. And
Mhm.
the first one is, why is Listeria ivanovi not detected by N-Light, and how is it different from other Listeria sensus triple group?
It's using a different enzyme target, and this enzyme is not expressed by Ivanhoe. But we're already working on a concept that in future Ivanhoe may also be included. But even now, as the test is now, we think it's still worth a while to use it because Ivanhoe will be there really rarely. And yeah, that's as much as I can say. The reason it's not like let's say the ISO method with respect to Ivanhoe is just a different enzyme target. But we selected this target because we wanted to have less false positives for such a rapid test.
Okay. And the second question from Gregor is, how long can a vial be transported after positive result to an accredited laboratory for PCR confirmation?
Um, Well, it should always be as fast as possible and transported cooled to the lab. This we have not really validated yet, but I would say like if it's analyzed within 2 days and cooled during all that time, should be fine. Um, yeah, there's still work to do to test out these things.
Thank you, Julian. The next question is actually there are more than just next question. I hope we will manage to answer everything in time. So, has the test method been accredited and is it acceptable to use for European and UK markets?
We have not yet um, done a certification or external validation for the test, but we plan to do it next year. Most likely, we will do an AOAC certification. So yeah, that's all I can say. I mean, we can provide like our internal data that we have collected in validating this test, and else also we can help if anybody would like to try it out. We can help in a kind of validation on site. Yeah, so that's all I can say about it.
Good. And like the second part of the question was about like, is it acceptable to use for European and UK markets? And I guess for sure, yes.
um, Yeah, yes, for sure, yes. You just have to be open about it that it has not yet this certification, but if customers show or users show that it works in their setting, for example, compared with another method, or then send then samples out for confirmation, I think it should be fine. Um, we see it also really as like more as a screening tool, so not as a replacement of all other existing methods, and then that it allows to do more sampling, more environmental monitoring, and to collect more data to do like informed decisions about where to improve cleaning and so on, to test before and after cleaning, maybe even without sending all of them out for confirmation. Uh,
Thank you, Julian. Next question is about specifically about data mentioned in your presentation. The strains that were not identified as Listeria spp in the study, what kind of microorganism were they?
we are working on that, and I will isolate some of them. It could be that maybe some Enterococcus, but it could be also some surprises, some other strains. So it's always with this enzyme-based, like with enzyme-based assays, you will always find some maybe which will give some positive results. Although we try to suppress most other species with our specific broths, but as the food factories are such a complex environment with so many different bacteria, it will never be that there will be not any false positives.
Yeah, unfortunately. Next question is, what is a false positive detection percentage of Listeria spp while performing with luminous methods or ICO or qPCR method? In general, how is it more reliable than qPCR or ISO methods?
To be honest, it will not be more reliable, but it's faster and then in the end also more cost efficient. You can do it on site, and you can collect like over the time data to see how, for example, really how well the cleaning is done and so on. So we are rather like advised to incorporate it in the scheme and not to use our test to replace all other methods, and but to use it as a screening tool.
Yeah. And is there any clarification about false positive detection percentage? Like if we have it.
That was in this one slide. So false positives was like of the total positives detected, it ranged like from to 30 to 60% or something. That would be like the false positives, but usually that will be just some other microorganisms.
Thank you, Julian, so much. And guys, if you have more questions, you still have time to drop it in. Me, I'm just curious, Julian, are there any restrictions towards the type of food factories who can use our test or not?
Until now, not really. So we have tested it in quite a range of different factories. Yeah, but it's really one has to try out, and then also we can advise always. We're happy to help and advise how to best use our tests, and then how to do later on follow-up analysis and so on. Yeah, we're happy to do that.
And
Until
in
now,
general,
we haven't found a setting yet where it's not at all working.
yeah, and in general, how would you define like the training process? Is it complicated or middle
Well, the
complication?
major advantage of this test is that it's really easy to use. So yeah, I think it can be learned within 10 minutes. And yeah, the instruments also are not really complicated. Yeah, so and also for the training, we are always happy to do some online training and also coming to the places and show the tests. So, but usually people will learn it very quickly.
Thank you. The next question is, have you compared the rate of false positive to esculin-based rapid tests or swabs?
The esculin test will have a much higher positivity rate, false positives, because you have lots and lots of bacteria which have beta glucosidase activity. And that's exactly why we chose this other target. We could have gone also for a beta glucosidase test, but we decided for this reason, to do it differently. Yeah, and for example, when we do like a Fraser confirmation, also in our studies, the ISO analysis, it's usually much higher. The tubes which will get black much higher percentage than in the end what will turn out to be Listeria SPP.
Thank you, Julian. Next question. Is this test also influenced by the turbidity of the broth, like the other NEMIS tests?
If it's affected by turbidity,
Yeah, probably yes, probably like this.
yeah, it depends. If the turbidity is coming from bacterial growth itself, it will not be that not have be that much of an effect. But if you have like a lot of food stuff which you transfer inside the test tube, this will be not good and may decrease the reliability of the test. So we always advise to avoid that and rather go like for seams and niches and so on where you might expect biofilm and so on. You can so we as I showed you in this presentation, we sampled during production or after production before cleaning, but also when we did it, we tried not to like have the full swap with lots of foods going in, and yeah, that's then usually it should work okay.
Thank you so much. And yeah, we have more questions. Actually, two questions came at the same time. What could be the cost of one swap roughly for luminous method? Like probably it's more or less raw test cost, right? If I'm not
I think that
mistaken.
you can answer, Alina.
Yeah, like for this question, I would suggest to write directly to sales@nemistech.com. So because the price varies, really, it varies from region as well. And the second
Yeah.
question, how we can ensure the cleaning or disinfection of equipment cross-contamination? Is there any mechanism present in equipment or any validation?
This I did not fully understand. So if there's cross contamination into our test from the equipment, or
Probably yes. So Raval, if you can just clarify your question, but as far as I understood it, like, is there a possibility to detect the cross contamination? In our test, I'm not sure that it's so
cross
AI-driven.
contamination from so I mean you can do sampling at two places and then see if you all get positives. But then to see if it's really the same strain or the same clonal lineage, you would have to do this follow-up analysis and do the whole genome sequencing and the isolation of the strain. But that's then possible with the test that you do this follow-up analysis. But it's not like direct. You cannot directly tell, okay, I've got this and this result with our test, and then I got it in another place, and then this positive must have come from cross contamination from someplace else. But you can identify if you recurrently find positives at one place or at one region, always before cleaning and maybe even after cleaning. That means something with the cleaning is not really good, and that means that's a high risk spot for even then in the end El Mono.
Yeah. Next question is, can meat surfaces be swapped and tested?
We advise against that, no, because in some cases that can be, if it's raw meat, there can be some endogenous enzymatic activity which can also play a role. So we do not advise it. So it's really a test for environmental monitoring and not for food testing.
Yeah, maybe here we need just to clarify again, like that our test is the best performing after cleaning surfaces, right? Here.
Yeah. So it's
So,
really good for like checking if the food contact surfaces are really safe.
yeah. So, guys, we have just like a few minutes left. If you have questions, you have last minute to drop in, or otherwise just text us in sales or tech support email. Uh, just question from me, Julian. Considering the recent recalls which occurred because of listeria, how do you think our test would help to prevent these outbreaks and recalls?
Well, it would be really a preventive measure because it's so easy to do and not so much effort. You don't need to send out and then wait the test and wait until you get the results. You can do more testing. You have more data, and then you can see the trends, and you can see okay, in this machine it's always in this edge, and then there might be some condensation water which will then drip onto the product and so on. That you see these issues and that you can fix them, and then and really to prevent that these listeria, especially L mono, will colonize your factory and form biofilms in specific places where there will always continuous contamination of the products which are produced there. I think that's the main value. It's the main value of environmental monitoring, and I think it's the way to go to really reduce the number of recalls. But yeah, we can only like encourage people to do this, to do environmental testing, whichever method is used. And I think our tests have some value there because they're so easy to use, and you can generate a lot of data points.
Thank you, Julian. And there came some more questions. One of it is, can we swap the surface of doormats and shoe cleaners?
Doormats and shoe cleaners. Yeah, probably yes. If you use the neutralizer, should be possible. Depend then how much of the disinfectant is there. Maybe that's something one can also check in the lab. Yeah, but in principle should be possible, or also at least around it. For sure, it should be possible to swap like shoes at the end of the before going to the disinfectant bath or so, and of shoes maybe before when people come in. Yeah, I think
Yeah.
could be such specific things we have not. That's hopefully by discussions with the users we will find out more, and yeah.
But in general, can you summarize where it's forbidden to swap at all? Are there any specific areas?
um, Forbidden, yeah, if it's like maybe you should not stick it in a very slimy sink and drain. That's always the question of overloading, and also not right into the food paste or so, but rather really on the surface around surface, wet surfaces and so on. Yeah.
Okay. We have just 5 minutes left. If there are no questions, guys, just shoot on more reactions. So we understand that it was really valuable webinar session because for us it's very
Yeah.
important. As the next important product we are very, very close to launch is a maxi sampler. But again, stay tuned for future updates. And don't forget to subscribe for future webinars from NEMIS Academy. Thank you so much, Julia. It was really
Yeah. Thank
valuable
you,
session.
Alina. Thanks a lot. Yeah.
Uh,
Okay.
sorry, one more question. Yes, it was very late, but I managed to catch
Yeah.
it. So,
Okay.
can Listeria grow in cold store of meat products where temperature remains minus 18 degrees Celsius?
It will not grow for sure, but for sure it will survive because they're very good in surviving all kinds of conditions. So then if the meat is thawed again, the listeria will still be there, but they do not multiply at -18.
Okay. Okay. Great. Thank you so much, Julian. Thank you, Raval, for last question. And now we are good to close and have an
Yeah.
amazing day, guys.
Yeah.
And
Have a
just
nice day, and thank you, everybody. Bye-bye.
bye.
Bye.
Hello.